Imagine a world where creatures thought lost forever suddenly roam the Earth again, defying extinction and rewriting what we believed about life itself. Extinction, once considered permanent, has now been challenged by science, revealing astonishing possibilities that blur the lines between nature and human innovation. From giant birds to tiny frogs, certain species have miraculously returned, making scientists and conservationists rethink the limits of revival and preservation. The woolly mammoth, extinct for thousands of years, now exists not just in fossils, but as a possibility in laboratories using modern cloning techniques. Some animals disappeared due to human activity, others vanished naturally, yet today, efforts to bring them back are reshaping the future of biodiversity worldwide. Consider the Pyrenean ibex, gone from the world in 2000, yet briefly returned thanks to cloning before fate intervened once again. Each resurrection story carries hope, controversy, and ethical dilemmas, prompting us to question whether humans should interfere with nature's final verdict. Scientists call this process de-extinction, a field that combines genetics, cloning, and deep ecological understanding to bring lost species back to life. While some projects focus on species with recent extinction, others attempt to resurrect animals vanished tens of thousands of years ago, like the mammoth. The excitement isn't just scientific, it's emotional, inspiring global audiences to imagine forests filled with creatures once thought forever gone. Some extinct animals are brought back to combat environmental crises, like restoring lost pollinators to help ecosystems survive. But de-extinction also raises moral questions. Should we bring back animals just because we can, or only when necessary for ecological balance? The technology behind this revival involves extracting DNA, editing genomes, and sometimes using close relatives as surrogate mothers for gestation. Despite immense challenges, these projects have made history, proving that extinction is no longer always the final chapter in an animal's story. The passenger pigeon, once numbering in billions, vanished within decades but inspired ambitious plans to reintroduce a species crucial to forest regeneration. Resurrection doesn't just capture human imagination, it offers a second chance for species to reclaim lost habitats and ecological roles. With each new attempt, researchers face unpredictable outcomes, from partial successes to tragic failures, testing both science and patience. Yet the prospect of seeing creatures like the moa bird towering over landscapes again sparks fascination and debate alike. De-extinction is not science fiction anymore. It's a tangible, controversial reality, merging biology, ethics, and hope for the planet's future. Beyond mammals and birds, tiny amphibians and insects once lost to disease or habitat destruction are now candidates for revival projects. Every recovered species challenges our understanding of extinction, evolution, and the fragile balance between human activity and the natural world. Advances in gene editing, like CRISPR, have made bringing back extinct species faster and more feasible than ever imagined in history. Some scientists aim to create hybrid species, blending extinct DNA with living relatives to recreate lost animals that can survive today's environments. But not every species is suitable for revival. Fragile ecosystems, limited resources, and ethical concerns limit what humanity can or should attempt. The stories of these revived creatures are not just scientific, they are emotional, capturing hearts, headlines, and imagination across generations. Imagine witnessing a Tasmanian tiger, extinct for decades, roaming wildlife reserves, reminding humanity of both loss and newfound hope. Some animals return only in controlled environments, highlighting the gap between laboratory success and survival in wild natural habitats. Still, each step in de-extinction represents a triumph of human ingenuity and curiosity over the irreversible forces of time. While critics argue that resources might be better spent on conserving living species, proponents see revival as a powerful conservation tool. The science is evolving rapidly, with techniques that were unimaginable a decade ago now producing viable embryos and early-stage creatures. Some animals come back thanks to frozen DNA, preserved tissues, or even amber fossils, unlocking secrets buried for millennia. The story of the Pyrenean ibex proves that cloning can temporarily succeed, but survival in the wild remains a monumental challenge. Meanwhile, the passenger pigeon story shows hope for species that disappeared too quickly for natural recolonization or adaptation. Scientists also study how extinct animals interacted with their environment, ensuring revived species can survive and thrive without harming current ecosystems. To replace a habit effectively, you first need to notice what prompts it, because awareness is the first step toward intentional change. Triggers can be external, like places, objects, or notifications, or internal, like emotions, fatigue, or boredom that signal your brain to act automatically. Often people mistakenly blame themselves for repeating a habit, but most habits are triggered by cues that happen without conscious thought. For instance, craving a snack at the same time each day is usually linked to a daily routine or environmental cue rather than hunger alone. Observing your habits without judgment helps identify patterns, making it easier to plan practical replacements that fit into your day. Many attempts at habit change fail because people focus only on stopping the behavior without understanding the underlying triggers. Recording when and where habits occur provides clear insight into consistent cues and the situations that prompt automatic behavior. You may notice habits appear at the same time, location, or in response to certain feelings or people. Once you recognize these triggers, you can plan new behaviors that respond to the same cues in healthier ways. Understanding the reward your brain receives from the habit is equally important because habits continue when the reward feels satisfying. A habit like scrolling social media may provide quick entertainment, while stress eating provides comfort, even if temporarily. Identifying the real reward allows you to substitute a behavior that provides a similar benefit without negative consequences. This approach emphasizes replacement instead of restriction, which increases success and reduces the feeling of deprivation. Paying attention to subtle cues, like emotions or daily routines, gives you control over your habits instead of letting them control you. Triggers can be visible, like snacks on the counter, or invisible, like habitual responses to stress, boredom, or tiredness. Emotions often trigger habits. Understanding your feelings in these moments helps plan alternative, productive responses. Once triggers are identified, you can experiment with replacing habits while keeping the same cue and reward structure. For example, instead of scrolling when bored, you could read a short article or take a quick walk to achieve similar mental stimulation. Planning replacements in advance ensures you have a constructive response when the habit trigger occurs. Tracking the context, time, location, and feelings associated with habits provides valuable insight for designing replacements that actually work. These observations allow you to understand which cues are most influential and prioritize them for habit replacement. Over a few days or weeks, patterns emerge, making it easier to predict when a habit will appear and prepare accordingly. Habit stacking is an effective technique here. Linking a new habit to an existing behavior ensures it occurs naturally when the trigger arises. For example, if you usually check your phone after lunch, replace that action with stretching or stepping outside for fresh air. The key is consistency, performing the replacement behavior whenever the trigger appears until it becomes a natural routine. Gradually, the new behavior begins to feel automatic, reducing the likelihood of reverting to the old habit. Environmental cues play a significant role. Arranging your surroundings to support positive behaviors can make replacement easier. For instance, keeping healthy snacks visible and moving unhealthy ones out of reach nudges your behavior in the desired direction. Similarly, setting up reminders or visual cues can reinforce new habits, prompting action without relying solely on memory or motivation. Identifying triggers and rewards equips you to make thoughtful choices rather than acting out of habit unconsciously. Understanding triggers shifts the focus from punishment or guilt to planning and creating practical solutions for habit change. Once triggers are clear, you can confidently design new routines, ensuring each action aligns with your goals and values. This approach removes pressure from willpower alone and instead leverages the brain's natural response to cues and rewards. Tracking progress and adjusting strategies as needed strengthens your ability to maintain replacement behaviors long-term. Awareness of triggers and rewards forms the foundation for safe, practical, and sustainable habit change that fits everyday life. By observing habits objectively, you gain insights into which actions need replacement and which cues you can leverage for positive routines. The more accurately you identify triggers, the easier it becomes to insert a replacement habit that satisfies the same underlying need. Structured observation, consistent replacement, and environmental design together create a realistic pathway to building better habits gradually. Habit change doesn't rely on motivation. Beyond famous examples, some extinct species are quietly returning, like rare frogs and insects, demonstrating that de-extinction extends beyond iconic animals. The gastric brooding frog, extinct since the 1980s, inspired scientists to experiment with tissue preservation to recreate its remarkable reproductive process. This frog uniquely swallowed its eggs and gave birth through its mouth, a biological marvel scientists hope to restore in modern ponds. Tiny in size but massive in ecological importance, reviving amphibians can restore balance, acting as both predators and prey within their habitats. Insects such as the Xerces blue butterfly, lost to habitat destruction, are also candidates for revival, crucial for pollination and ecosystem health. Scientists extract DNA from preserved specimens, sometimes decades old, piecing together genomes like a complex puzzle of life itself. Stem cell technology allows researchers to create embryos carrying extinct traits, bridging the gap between preserved DNA and living organisms. Even birds with lost migratory patterns can potentially return if revived, thanks to careful genetic and behavioral guidance by conservationists. Hybrid approaches combine DNA of extinct species with closely related animals, producing functional surrogates capable of surviving in modern ecosystems. The quagga revival demonstrates how selective breeding guided by DNA can recreate animals visually similar to those long vanished. Beyond visual resemblance, scientists focus on ecological function, ensuring revived species can fulfill roles once lost in nature. One lesser-known project aims to revive the heath hen extinct in the 1930s using preserved DNA and close relatives like the prairie chicken. These efforts highlight that de-extinction is not just about the largest or most famous animals, but about restoring ecological balance. Cloning remains difficult. Often embryos fail, gestation is complex, and newborns may struggle to survive, underscoring the limits of current technology. Nevertheless, each attempt yields insights, from genetic viability to ethical considerations, shaping the future of species revival. Modern gene editing tools like CRISPR allow unprecedented precision, enabling scientists to modify DNA sequences to match extinct species more closely. This technology has opened doors previously unimaginable, making the resurrection of species extinct for thousands of years theoretically possible. Some projects even explore recreating lost traits, such as cold resistance in mammoths, to ensure survival in harsh contemporary climates. Revival is about more than curiosity. It can reverse centuries of ecological damage, restoring missing links in fragile ecosystems. Each revived animal must adapt to modern habitats, which have changed dramatically since their extinction, presenting a new challenge for conservationists. Researchers study historical records, fossils, and preserved specimens to understand behavior, diet, and habitat needs of extinct species. Even small mammals, like the Florida panther's extinct relatives, are considered for revival to maintain genetic diversity and ecosystem resilience. De-extinction efforts often intersect with endangered species programs, reinforcing the idea that saving one can complement the other. Public fascination drives funding and support, turning scientific laboratories into hubs of education and wonder for people worldwide. Some revived species act as keystone organisms, restoring ecological functions such as seed dispersal, soil aeration, and predator-prey dynamics. Scientists approach revival cautiously, balancing innovation with ethical responsibility, ensuring new life does not create unforeseen consequences. Each project provides insight into evolution, revealing how species adapt, survive, or disappear in response to environmental pressures. Even extinct plants are being revived, complementing animal restoration by recreating lost habitats and supporting food chains. Ancient DNA extracted from amber or frozen soil provides genetic blueprints, unlocking possibilities for species thought forever lost. Behavioral studies ensure revived animals can thrive, For example, teaching birds to migrate or predators to hunt safely in controlled environments. Conservationists debate whether revival should focus on ecological benefit, species importance, or public fascination, reflecting the complexity of decision-making. Many lesser-known amphibians face extinction due to disease, and revival projects aim to combat this while restoring species diversity. Scientists use surrogate species to carry embryos, a crucial step in bridging extinct DNA with living ecosystems. The process is painstaking, requiring years of research, trial and error, and immense collaboration among international teams. Advances in cloning and gene editing have made it increasingly likely that extinct animals will live again, at least partially. De-extinction sparks heated debates. Should humans resurrect species we drove to extinction, or focus instead on protecting endangered animals still living? Critics argue resources for revival could divert funding from urgent conservation projects, leaving at-risk species without the protection they desperately need. Supporters counter that revival can complement conservation, restoring lost ecological functions and giving extinct species a second chance in managed habitats. Ethical concerns extend to animal welfare, as cloned animals may suffer health problems, deformities, or shortened lifespans, raising moral questions. The Pyrenean ibex demonstrated this vividly. Its cloned return lasted only 7 minutes, highlighting the fragility of revived life. Some worry that revived species could disrupt modern ecosystems, outcompeting native wildlife or introducing diseases to which current species are vulnerable. Others question the human right to play God, manipulating life and death for curiosity, or ecological experiments without fully understanding consequences. De-extinction blurs lines between nature and technology, raising philosophical questions about whether humans should undo extinction caused by their own actions. Critics argue that focusing on charismatic species, like mammoths, may skew public perception, leaving less appealing but ecologically crucial species neglected. Supporters emphasize ecological restoration, showing that reviving keystone species can rebuild ecosystems and benefit countless other organisms. Genetic engineering presents dilemmas. Altering DNA to recreate extinct traits may yield hybrid species rather than true originals, challenging definitions of authenticity. Even small missteps can have cascading effects, potentially harming ecosystems already stressed by climate change and human activity. Critics caution that novelty alone shouldn't justify revival. Decisions must prioritize ecological benefit, ethics, and long-term survival prospects. In some cases, revived animals may survive in laboratories but struggle to adapt to natural habitats, questioning the feasibility of reintroduction. Conservationists stress that proper habitat restoration must accompany revival, ensuring animals have the resources and space to thrive. De-extinction also raises legal questions. Who owns revived species, and who is responsible for their protection and welfare? International regulations are still catching up, leaving gaps in how revived species are managed across borders and ecosystems. Public perception plays a role. Revived species often garner excitement, but enthusiasm must be tempered with understanding of ecological responsibilities. The moral weight of bringing back species extinct due to human activity highlights the importance of accountability in scientific endeavors. Critics argue that focusing on the past may divert attention from preventing future extinctions, which could be more cost-effective and impactful. Proponents highlight hybrid approaches, showing that partial revival can restore ecological functions even if the exact species isn't recreated. Ethical frameworks guide revival projects, balancing human intervention with animal welfare, ecosystem integrity, and long-term sustainability. Some scientists advocate functional resurrection, prioritizing ecological roles rather than exact species identity to maximize environmental benefits. Controversies also arise around genetic modification. Editing DNA for survivability may conflict with natural evolutionary processes. Wildlife organizations emphasize caution, advocating pilot programs and controlled environments before broader reintroduction into wild ecosystems. The possibility of failure is high. Cloned or hybrid animals may not reproduce, adapt, or survive, risking wasted effort and resources. Revived species often require intensive care initially, including habitat management, disease prevention, and behavioral guidance. These projects challenge humanity to balance ambition with responsibility, navigating the delicate interplay between science and ethics. Critics warn that revival could create complacency, making humans less motivated to prevent extinctions of currently endangered species. Proponents counter that revival can inspire conservation awareness, showing that human ingenuity can correct past mistakes and restore lost biodiversity. The cost of de-extinction is significant, spanning research, cloning, habitat restoration, and long-term monitoring, making prioritization essential. Some argue for focusing on species with recent extinction, where DNA quality is better and ecological conditions are easier to recreate. Others push for ambitious projects, like the woolly mammoth, aiming to demonstrate what science is capable of achieving on a grand scale. Ethical debates also touch on animal identity. Are hybrids truly the same species, or a human-created facsimile with no real contin? The final step in replacing habits effectively is adopting a mindset that supports gradual, consistent, and realistic improvements over time. Seeing habits as part of your identity helps make change sustainable because behavior aligns naturally with how you view yourself. For example, thinking, "I am someone who moves daily," encourages exercise without relying only on willpower or external pressure. Focus on small, consistent wins rather than trying to overhaul everything at once, as incremental progress is more achievable and reliable. Curiosity replaces judgment. When you notice a slip, explore what triggered it and consider how to adjust instead of feeling guilty. Self-compassion reduces stress and helps you stay consistent, reminding you that slips are normal and part of learning new routines. Treat habit change as an experiment. Observe what works, what doesn't, and refine your approach gradually for the best long-term results. Environment matters. Arranging spaces to make positive habits easy and less visible temptations hard to reach supports consistent behavior. Tracking progress reinforces habits by giving visual evidence of success, which motivates repetition and strengthens the behavior over time. Celebrating milestones, even small ones, provides a reward that encourages the habit to continue without relying solely on motivation. Planning for challenges ensures habits remain consistent during busy or stressful periods, making setbacks easier to manage without discouragement. Pairing habits with enjoyable activities makes routines more appealing, such as listening to music while exercising or reading during a break. Identity alignment strengthens habit adoption. Every time you act in line with your values, the behavior becomes easier to repeat naturally. Mental rehearsal is helpful. Imagining yourself performing the habit successfully in challenging situations prepares your mind for real-life execution. Flexibility is important. Adapt your strategies when circumstances change, ensuring that habits remain realistic and achievable in different contexts. Patience is essential. Habit change takes time, and consistent, intentional practice gradually builds routines that feel natural and integrated into life. Focus on progress, not perfection, and view small improvements as meaningful steps toward long-term positive change. Observe your triggers and rewards continuously, adjusting habits as needed to maintain alignment with your goals and values. Over time, replacement habits become automatic, requiring less conscious effort, and old routines fade naturally without forced restriction. Your brain can form new positive routines when guided with awareness, consistent practice, and supportive environments. Every small, intentional action contributes to shaping your behavior and reinforcing the identity you want to embody. Consistency, planning, and self-reflection turn habit replacement into a sustainable process, making positive behaviors a regular part of daily life. By focusing on gradual change and supportive systems, habits become easier to maintain, even during busy or challenging periods. Celebrate the process, not just the outcome, as each day of consistent practice strengthens your ability to maintain new routines. Habit mastery is less about motivation and more about designing systems, practicing consistently, and aligning behavior with identity. With awareness, planning, and persistence, replacement habits naturally integrate into life, supporting goals, well-being, and daily productivity. When setbacks happen, treat them as opportunities to learn, refine your strategies, and reinforce the habit loop with small, achievable actions. Over weeks and months, positive habits compound, gradually reshaping routines, lifestyle, and how you respond to triggers and challenges. Focus on creating an environment, identity, and systems that consistently favor good habits, making them easier and more automatic over time. Understand that replacing habits is a process. Progress builds gradually, and sustainable change requires consistency, reflection, and adaptation. With the right mindset, replacement habits become part of your routine, supporting personal growth and helping you make consistent, intentional choices.